welcome everybody. We've finally made it and got together, so that's nice. Now, for our audience today, let's introduce everybody, and we'll start, of course, with the ladies first. Anne-Marie, you're from overseas, so would you like to just introduce yourself a little bit?
Sure. Thank you, Brian, and thank you to UK Calm for. So my name is Anne-Marie. I'm a PhD. I'm French. I'm a French citizen, although I was born in Cambodia. So I have a PhD in organic chemistry from Montpellier, University of Montpellier, and so I'm an organic peptide chemist expert, as to wish. So protein expert. And I did my PhD on radical reactions on amino acids and synthesis on the dihydroamino acids using polymers. And that's why I understand the magnetofaction PAG virus that they did, the technology that they're using. And then I did my postdoctorate in United States in University of Michigan. My first postdoctorate was identifying the membrane proteins using 2D gels and mass spectrometry. And my second PhD was studying the immune process inflammation in RA, rheumatoid arthritis. So, and I live now in Luxembourg, and I have been opening my own structure office in 2018, and I'm tutoring science and language to children from primary school to high school and also to university.
Okay, Anne-Marie, thank you for that. I think it's right to say that in your particular fields of work and the research that you've done, you count as a world expert.
Yes, of course. I have been working with the best expert in the world. For example, my lab was in University in Michigan. It was Ann Arbor, and it's top 10 out of the 200 university. And at that time, the proteomics technology were only developed in 5 labs only. Now it's everybody has his sequencer or his HPLC or his mass spectrum. At that time, we were pioneers. We were pioneers in the field. So yes.
Okay, right, lovely. Thank you very much. And you're speaking to us from Luxembourg this evening.
Yes, that's right.
Okay, all right. And Debbie, we've done quite a few things together, but now is your opportunity to tell the audience a little bit about yourself.
Very quickly, I'm a retired nurse, National Health Service trained. I trained in at the Royal Free back in the 70s, and I've done a whole range of different nursing courses, ranging from spinal to ear, nose and throat. I was a sister on my own ward in London, then had a family, and I've been doing a lot of research since, especially into one other virus, an RNA virus, hepatitis E. So, and I'm generally quite nosy, and I love to research. And yes, we've been doing quite a lot of work together and seeing what's been making the difference and what the NHS was in my day and what it meant to nurses and doctors, and what seems to be happening in today's world with regards to the whole health system and to certain things that are going on, asking questions.
Okay, I'm going to say, Debbie, you've actually achieved some quite remarkable things, because by your own research, you've highlighted a lot of really key points around, particularly the MHRA yellow card system. We've had a number of discussions about that. So you highlighted a lot of discrepancies, inaccuracies, and just generally strange reporting in that yellow card system. And we've done some specific discussions on that, which have aroused a lot of interest. But it was you listening into a Telegram group, which has got you in contact with Anne-Marie Yim. Just tell us a little bit how that happened.
Well, I mean, as everybody can see, I'm a bit of a silver surfer, so I'm not terribly up on technology, and I've got Telegram, and I message people, but I don't really know how to use it. And all of a sudden, I saw a chat that was taking place on Professor Dolores Cahill's page, and I didn't really understand what I was doing, and I hit the button to join the meeting, and to join the discussion, and to listen into what was being said, and I could hear Anne-Marie and Dr. Och talking about their experiences and their discoveries and what was happening in France and Luxembourg particularly. And it's kind of fitted in with a lot of the work that we were doing already, Brian, with the whole genomic industry and bio industry that we featured on the latest No Smoke. And so I thought I'd give it a go and try and contact Anne-Marie on Telegram with the video from UK Column, the PCR and the Genome Beast. And much to my delight, and I'm very privileged that Anne-Marie, I mentioned a couple of other things, which I'm sure Anne-Marie will explain. It involves a substance called peroxynitrine. And I threw it Anne-Marie's way because she's the expert. And at the end of the day, I'm just a nurse and it's not my area of specialism. And that's where we are today. Very kindly, Anne-Marie contacted me and then in turn you. And now we find ourselves here today talking about all of this.
Okay.
I think, if I may say, I'm very privileged to know both of you, and I think it's a honor. And you're wonderful people, and especially Debbie, and I call her Miss Marple. You know.
Yes, due to all of her detective work and her research. Okay, thank you very much. And we've also got Dr. Harvey Seligman with us this evening. Unfortunately, Dr. Seligman is only going to be audio, but can you hear us okay, Harvey?
I hear you well.
Okay, please just introduce yourself and give us a bit about your background and your particular expertise.
Well, I was born in Luxembourg. I did a master thesis in plant physiology in Jerusalem at the Hebrew University, and a PhD thesis in vertebrate morphology, also at that university. And after that, I did a number of postdocs, more or less on molecular evolution and on bioinformatics. And I've been an invited professor for 5 years in the unit of Professor Didier Raoult in Marseille from 2013 to 2018, where I did mainly molecular biology studies.
Okay, thank you very much for that. So you're another eminent person in your field, and we're
no,
privileged.
The field doesn't exist without me.
Excellent.
Yes,
Good. Now we know exactly where we are. For anybody who doesn't know me, I'll just say that I'm part of the UK Column team. I'm one of the regular presenters of UK Column News, and as well as that, I do a lot of work researching, quite a few different subjects, but it just so happens that, of course, over the last 18 months, it's been COVID and matters to do with vaccine adverse reactions and lockdown and removal of liberties that has been the important factor for me. And I've been very pleased at the response we've had from our wider audience to various discussions that we've broadcast. So my job tonight is very simple, because the subject matter that we're going to cover around COVID-19 and vaccinations, I'm going to say, is way beyond my understanding. These are very complex scientific fields, and so my prime job, I think, is just to bring the discussion together in a way that the ordinary person can really understand what's going on. So my job is going to be a little bit of being a referee, and I'm going to try and make sure that you highly qualified scientific people are going to get the discussion into language that us ordinary people can understand, because what we're trying to do here is to take some quite difficult concepts and put them in a form that we can share out to a very wide audience. So I'm going to say I'm going to be a little bit of a referee, but I'll try and do it nicely. Okay, well, thank you very much for those introductions. I'd set out some questions earlier, which I'll stick to. We'll work through those, but I just took the questions because they were things that seemed to be important to me, and I felt that if we could work through them in a fairly clear and organised way, this would make it much easier for the audience to understand what, as I've said, are obviously some quite difficult subjects. So I wanted to begin somewhere very simple and say that over my lifetime, Debbie's lifetime, we're going to say that we've experienced flu. So flu's been a natural part of our life because it tends to crop up every winter. I have never had problems. Perhaps I've had it once or twice maybe in my whole life, but in general I haven't suffered any problems. But we know flu does come along, and we know that when the flu season starts, it can be quite dangerous to people who are very elderly, and certainly to people who are elderly and have underlying health conditions. And we know that unfortunately in UK, in an average winter, about 40,000 people die as a result of flu, whether that's coming in on top of other complications, but nevertheless, that's the statistic. And that's a statistic that most people are completely unaware of. If you said to them that number of people die every year, they would be quite shocked. But the fact is that that is a perfectly normal figure. But in this case, we've got COVID that suddenly appeared, and we're being told that COVID was very different, that COVID was dangerous, COVID needed special precautions. So if I can throw it back to you in the first instance, Anne-Marie, what is all this talk about COVID? What is COVID-19, and why have we been told that it's so dangerous?
So yes, so the COVID pandemic, as you all know, started in Wuhan in November, December 2019. That's when everybody learned about Wuhan and that the city has been quarantined, and that people were sick from this disease. So coronavirus, called coronavirus 19 because it's appeared in 2019. Coronavirus because like the flu, it's a coronavirus. But this one is different from the others in the sense that it's bigger and it seems to have been bioengineered, like man-made. So first they made us it was a man-made virus that originated from bats, and that then it jumped from the pangolin, and that somehow it infected man. But we can see that this narrative is falling apart because of Dr. Fauci emails revealing that it was actually a research on game that has been funded by the NIH in the P4 laboratory of virology in Wuhan. So this coronavirus clearly is quite dangerous because it's called an artificial virus. It's a chimera virus. So it's big. It's 32 to 36 kilobase. And it has a particularity, like it has 4 inserts of S1 protein on the surface. And these S1 proteins can also be found in the HIV virus. So, and it enables the protein to enter the surface, the cells, by binding to the ACE2 receptor, the angiotensin receptor. And then it can be transmitted by aerosol ways, meaning by just breathing it, and then it infects your lungs. So, and it causes this acute respiratory disease. and usually from day 1 to day 5, you have mild symptoms like fever, cough, and difficulties to swallow, or maybe you lose the ability to smell, and then the disease evolves very quickly from day 5 to day 12, and where you can have more severe symptoms. It means you can't breathe anymore. You have inflammations of the lungs, and then you have this water going on into your sacs, your alveoli. How do you say?
Alveoli.
Alveoli, yeah. And then, of course, you need to be brought to ICU to be given oxygen because you cannot breathe anymore. The exchange between oxygen and CO2 doesn't work anymore. And at that time, when you have that, you can have a infection by bacteria, and you can have a sepsis, and then you can, of course, die. Yes, and it can be brutal. So from day 12 to day 14, if you're not treated correctly, you can die because your lungs don't function properly. So it's an organ failure, and then you die. So,
right. And you're being very firm on the fact that this is a virus that has been enhanced.
Of course, because as you all know, the Dr. Fauci is the director of the Allergy and Infectious Institute in United States, has been heard by the U.S. Senate, and they have been releasing his mail from January to March 2021, and we can read on some emails that there is a protocol of engineering these gain of function research. So it's clearly established now that United States has been founded through NIH and Dr. Fauci, and also through Bill Gates, this gain of function research, which is still going on right now in United States in 11 laboratories. So that's very, very disturbing. And so they had already these results in 2015, because this woman, this PhD, her name, who was leading the laboratory of virology in Wuhan, they published it. They said that they know how to make a virus infect human and cross the barrier of species, which it shouldn't normally happen.
Right. And how can I ask you how you think how did the virus spread in the world, appear in the world, and then spread?
Yeah, that's so. We have there is a professor in France. His name is Professor Jean-Bernard Fourtillon. So he's a chemical engineer, and he was a professor, and he therapeutic chemistry to the University of Medicine and Pharmacy in Poitiers. And why is it relevant? Because he has been jailed since April. He's 77 years old right now, and he has been jailed since April 2021 in Paris. And they release him. They have no charge against him, but they don't want to release him because he has been investigating on this virus, and he has been showing that there is research has been done, namely the research on the insert, on the S1 protein insert, has been done by a man. His name is Dr. He's a PhD also. He works from the INSERM. INSERM is Institut National de la Recherche Médicale. So it's like the equivalent of the NIH in the United States. So it's a health institute, public health institute research, specialized. It's a unit which is specialized in virus and bacteria, etc. So he has documented this very well, and he has shown that the Pasteur Institute in Lille, they hold 5 patents on the SARS-CoV-2, and the scientist who did the insert was one of student of Professor Luc Montagnier. So Professor Luc Montagnier, he's a Nobel Prize who discovered the HIV virus. He got the Nobel Prize for that, and he has been working on the mechanism of infection. So how once the people are infected by the HIV, thanks to the S1 proteins, how their immune system is being shut down. So that's why it is relevant. And you have to know that people in the research world are collaborating. It means that they are exchanging not only but also materials and also people. Like they send people from one lab to acquire the technology, and they are collaborating. So it's a very common thing to collaborate. And for example, France has been sending 300 scientists in the Wuhan lab from 2017 to 2019. And how do we know that? It's because once our Ministry of Health, Madame Agnès Buzyn, she once the lockdown has been done in December 2019, in March, in France, 19th of March, they started the lockdown, and she has been sending a military plane to get back the 300 scientists from the Wuhan lab, and it was through a military plane. Whereas the flight has been closed. The flights have been closed France to China, and the Wuhan city has been quarantined. So nobody should be able to be getting out or getting in. But yet there was between 500 and 1,000 French citizens in Wuhan, among them 300 scientists, and they managed to be brought back in France. And that tells me that something is wrong. Plus, in 2017, the husband of Madame Agnès Buzyn, which his name is Yves Lévy, and he was at the head of the INSERM. And he, in 2017, along with Bernard Cazeneuve, the Interior Minister of France, to inaugurate the P4 laboratory in Wuhan. And this laboratory, there's very few labs who can't do research, who are labeled P4. It's in Lyon, le laboratoire Mérieux. So they made a copycat of, if you wish, of the P4 laboratory of Mérieux in Wuhan, with the same facilities, etc. So it's obvious that there was a clear collaboration between France and China and United States and China. So that's clear.
Anne-Marie, so come back to this question then. There was collaboration. We got France and America and the Chinese working together with the lab, with this duplicate lab established in Wuhan. How did the virus actually get out? Do you think it was a mistake, or do you think it was...
I don't think it's a mistake. I don't think. Why do I say that? Because there's this book. You know, the book. It's called "The Eyes of the Darkness," where it was published by Dan Kunz in 1981, and it's a fictional book who talks about a pandemic and that this virus has been bioengineered in Wuhan, in China, and it has been released to create a pandemic. So is it a coincidence? I I don't know. I don't think it is. I think there's and then there's this Davos meeting with Klaus Schwab and Bill Gates, who have been rehearsing during their they have a scenario, and they have been studying how would the virus spread, and how it will advance and kill people. So my understanding is all these simulation and all these things are not random. And plus, there is also another meeting which has been reported by the French journalist, where there was a meeting between Bernard Kouchner, so an old minister of health, and the head of Glaxo, of Boiron, and of there was basically it was a meeting, a university meeting between many successive health minister of France and directors of vaccinations laboratories like Sanofi, Glaxo, Boiron. So all those laboratories who are manufacturing, so you wish, the vaccine, because not all pharmaceutical companies do the manufacturing of vaccines. It's like Sanofi, Pasteur, Boiron, Glaxo, Pfizer, and now, of course, AstraZeneca and Bio. So there aren't so many big pharma who are able to have the technology and are able to manufacture it.
Yes, okay. Well, we're going to come on to vaccines in a minute. My next question is that the COVID became apparent. There was a pandemic declared, and we were all told to be very frightened. We were told that a lot of people were going to die, a lot of people were going to become very ill. But as things unfolded, that didn't seem to happen in UK. We built massive hospital bed facilities, the Nightingale hospitals, for thousands of beds. And we know in the event that only a very, very few people went into them. I think in one, not even 100 or a few hundred in a multi-thousand bed facility. And we know from talking with consultants, nurses inside the NHS in UK, that many of the specialist COVID wards never received any COVID patients at all. So the government said that it was going to be very dangerous, that a lot of people were going to suffer with COVID-19, and indeed vulnerable people were going to die. But in the event, this didn't seem to happen. Debbie, I don't know whether you'd like to add a bit on that. We're talking about what we've seen as this so-called pandemic has unfolded.
I was actually talking to somebody about that today, who was recounting some of the images that you saw in Wuhan, people dropping to the ground and great big machines and loads of people in the streets using disinfectant and everything. And we really haven't seen that. And when the UK variant sprung to life, I think the rest of the world might have been looking at the UK thinking that, you know, there was bodies everywhere and ambulances flying around, and then it really wasn't like that. So the narrative that the government is giving and the narrative that the mainstream media are putting out isn't matching what people are seeing on the ground. Personally, I don't know anybody that has actually died of as a primary cause of COVID. I know of people that have died with and have had it on their death certificates, but primarily they've died of maybe a heart attack or a stroke or cancer, and with COVID as an underlying condition, not the other way around. So I'm finding a lot of people are now extremely confused because they've followed all the guidelines that the government's put out. Some of them have had 1 and 2 vaccinations, and they're still not allowed any freedom. So we're not seeing a pandemic, but we're acting as though there is still one going on.
So I wonder whether I'm going to come over to Harvey Seligman in a minute, but Anne-Marie, just you initially, what is your response to this? If we say to you that we've had the media just full of the fact that we are suffering a deadly pandemic, but the picture on the ground just doesn't compute with this, and the statistics don't seem to agree with this either. We know, and within the UK column, Mike Robinson has done, and the team have done quite a lot of work on the statistics through the COVID pandemic. And we're actually seeing that most of the deaths were caused as a result of locking particularly elderly people up in their care facilities. So very vulnerable people. We're seeing an increase in deaths, and we're seeing an increase in deaths of people at home. But we didn't see the increase in deaths due to COVID. How would you respond to that?
So I would say that they didn't imagine about the replication of the virus. I think they didn't anticipate the replication because the virus is so big, its replication is very, very difficult, and it didn't replicate as much as it would once it would infect a cell. That's one thing. They have shown that the replication stops within once the cell is infected because the size of the virus is so big. That's one thing that they didn't anticipate. That's why they had, my understanding, to release each time another strain. That's what I understand, because I read that there's a plan to release a 2019 virus and then load it with malaria. They can't insert whatever, they can't load it. They have the structure, like they have the structure to release it. And then on the internet that they want to release another one in 2021 and another one in 2023. So if they need to release it, it means like this virus, after a while, stops from spreading. Once you get infected and you recover, then you build an innate system in your memory, in your bone marrow, in your B cells, that is protecting you. So that's one thing. That's what epidemiologists are observing. They always, you see these curves where they have a peak like this, and then it stops. So you have a peak of mortality, and then it stops. The lockdown or the vaccination is doing what? You have a peak that is like this, like it's spreading more, but then it stops. Why does it stop? Because once you get infected and you recover, you acquire this herd immunity. And this herd immunity is not achieved through vaccination, but the fact that everybody got infected and build the immune system. That's that's what I understand. The second thing is like they have been exaggerating the whole thing. That's true, with the lockdown. And what is ridiculous is the guidelines. Like Dr. Ox, and not only him, but everybody in Canada or in France, everybody has said that these guidelines were ridiculous. Why? Because people were asked in Luxembourg alone during the lockdown of March 2020. They have been asking the physician to not work, to close their practice, to COVID military centers at that time, or hospitals. Each time we see that militaries are very present. So I'm just wondering if this war is just a bioweapon war conducted by the military through our own government. That's my question, because each time militaries are very much involved. And like in France, they have been, they talk about seven very sick people in the Metz, in the city of Metz, who were in the military hospital, and they said that they have nowhere to treat them. So they had to send them, like in Marseille and to other centers. And somehow they send one of those very sick people through a helicopter to the hospital of Ettelbruck in Luxembourg. And I was thinking, why are they sending these sick people like COVID positive and who declares a severe form of disease from Metz through helicopter through Ettelbruck? And I'm thinking, like, they don't have a hospital to heal them in France. So my understanding, it was like a spreading of the virus thing going on here. So, but that's my own opinion. The official narrative is they didn't have the facilities to treat them, so they had to treat them in Ettelbruck. And it doesn't make sense. And it was conducted by the military. So it doesn't make sense to me. And the thing is, so they ask the physicians, Dr. Ox told us, they ask the physician to close their office and then to come work 48 to 72 hours COVID military centers and the hospital, and then they were forbidden to do anything. They were given, they gave patients a box of paracetamol, and they had to send them at home for 10 to 14 days until they had complications. They couldn't breathe anymore, because now we know that paracetamol triggers inflammation and for severe death, severe COVID death. And then they will send the ambulance to pick them and to bring them in ICU and to put induced coma and to treat them with oxygen. And once in hospital, they were forbidden to give heparin, which is an anticoagulant, and an anti-inflammatory, and aspirin coagulant. So those guidelines, which is totally absurd, were given not only in Luxembourg, but they were given in France and they were given in United States. So they were given everywhere. So it seems that it has been coordinated.
Okay.
It has been coordinated.
Yeah, Anne-Marie, that's an extremely interesting, well, you've raised a number of interesting points there, but the key one that struck me was that you said that essentially the virus had not spread as quickly as you believe somebody hoped. This virus was supposed to be very potent. It was supposed to spread very quickly. And yeah. If I've got this right, you say this is partly because this is a very big virus. And
yes.
I think you said 36 kilobytes.
32.
What is that? When you say it's 36 kilobytes of what?
No, kilobase.
Okay.
So it's a sequence. It means like the virus, the core virus is either an RNA virus or an DNA virus. In this case, RNA virus, it's a single strand. And when you sequence these gene, you can know how many bases it has. And it gives you the size. It's nucleotides. Okay.
Right. Thank you for that. Now, I wonder whether we could bring in Harvey, Dr. Seligman. I would like to ask you for general comment on what happened when we were being told there was a pandemic. What did the statistics show us that was happening, as opposed to what the governments were claiming was happening? Are you able to comment on that side?
Well, the initial pandemic, the official numbers are much lower than the numbers that we got, for example, in this winter, maybe 4 or 5 times lower. So the initial panic was based upon very low numbers. Now, it's possible that these numbers were lower because there was less testing at the beginning. That's very likely. And an indication for that is that because there was less testing, the people who died were tested. So we had a higher percentage of mortality, not necessarily because there were more deaths proportionally, but because there were less people tested and only the very grave cases were tested. I don't have an answer on that. It's certain that when you have a pandemic or any epidemic, the virus or the bacteria starts frequently being very aggressive, pathogenic, lethal, and then lethality is something that is counter-selected in any population genetic scenario for a pathogen. And so with time, the very lethal ones disappear, and the less lethal ones, lethal, less mortal ones get more frequent. And that's the overall pattern that you see. It's not a linear pattern, but normally if you do nothing, with time, pathogens become less aggressive. They become, in principle, more contagious and less lethal.
Okay. A simple thing that we've noticed with the statistics in UK is that the government, or normally if the government is talking about people who are infected or have died as a result of a flu, those statistics are collected over a winter. And you talk about the statistics of a flu epidemic over a particular winter. And then we come into the better weather. Even in England, we get the better weather, and the flu disappears. And then the next winter comes, and then the new flu season starts. And the point I'm making is that the statistics are normally collected over one winter flu season. But for COVID, it is widely reported where the statistics are shown in a cumulative form. So we've moved on to the second year, and the statistics are being added together to the first year. And of course, this appears to magnify the whole problem for people who are not aware of how the statistics are normally collected and formulated. This is very deceitful, because the statistics should stand for each winter flu season, but for some reason with COVID, they're being brought together in a cumulative form. Are you able to comment on that? Why would the government do that, other than they're attempting to make it appear more serious than it is?
These are speculative questions. I usually don't talk about these things. That's the first thing. So I'm not specialized into the UK numbers. I looked a lot into the Israeli numbers, among others, because of family connections. So, but I can tell you there could be games about decreasing or increasing PCR cutoff levels, so that at some points you can artificially increase the number of positive cases, which would include more false positive cases, and vice versa, according to your politics, such as just before or after elections and so on, as happened in Israel. So and that's only one case. Or if you want to say the vaccine is working or not. So I'm certain there are manipulations. When I analyze the data, I do as if I simply believe the data that I'm given, that are published. And usually I succeed to show that with data, in this case, published by the Israeli Ministry of Health, saying, oh, the vaccine is working, I was able to show, no, it's the opposite, the same data. And the way it's done is they say, oh, look, unvaccinated people, 5,000 deaths, and the vaccinated people only, let's say, 50 deaths. But so the absolute numbers, they they assume that people will stop there, and I didn't stop there because the sample sizes were absolutely different. So 50 deaths among 5,000, that's like 1%, and 5,000 deaths among, I don't know, 1 million or 2 million. So in the end, it was 0.5% dead among the unvaccinated versus 1 or 2% dead among the vaccinated. Then if you take into account the duration of the time, because deaths accumulate over time, that's basically part of your what you were saying. So you get to a result that the vaccinated are 10, 20 times more likely to die per day per person than the unvaccinated. But that's a calculation that is not difficult. It's a percentage calculation. Anyone can do it. You don't need to have a PhD for that. And but the same data are used to say the vaccine is working.
Right.
And the basic cost benefit analysis, to tell you all, is like, if you say over 1 week, the vaccinated die 30 times more than the unvaccinated. So you need 30 weeks of perfect protection after some time. Let's say that the vaccination finally kicks in and actually protects people. You would need 30 weeks to be able to compensate for the excess of deaths. When I did with the real numbers, this calculation, I reached the point that there was a need for something like 700 days in total to compensate the excess death that happened just upon vaccination, meaning after the first dose, until the second dose, and in the first or 2 weeks after the second dose. This period is a period where people are very sensitive. The immune system is weakened, and therefore people are twice as likely to catch COVID. And when they catch COVID, they have a much greater risk of dying of COVID. So the risk of developing COVID is twice, and the risk of dying if you develop COVID is, according to the precise period of the vaccination, you're 10 or 20 or 30 times more likely to die.
Okay, thank you for that. Now, you have got some specific statistics which you have been working on. You've kindly produced some slides. If you're happy, we'll just leave those for a minute, and then we'll come back in and invite you to take us through the detailed work that you've done. I take your point that you didn't know I was going to ask you that generalized question about the statistical data on COVID. So thank you for answering that. But I just felt it was important because for ordinary people in UK, it has been apparent that something is strange in the way that certainly the British government's been handling data. Debbie, I wonder whether you can just comment a little bit on the anomalies that you've discovered with the collection. I know it's the subject of the collection of vaccine adverse effect data, but you found some strange things going on in the way the government's been handling the adverse reaction data.
Very, very strange, to the point where, I've contacted the MHRA, who are, at the end of the day, that's where the buck stops with regards to statistics and giving out the statistics via uk.gov. And we're seeing some, it would appear that even though this system in the UK is meant to be artificial intelligence, it's meant to be state-of-the-art digitalization, where NHS now is NHSX, digitalizing it, you would have thought that the serious adverse reactions, when they're reported, would be reported accurately, and that there wouldn't be any mistakes, because that's what computers are meant to do, isn't it? Not make mistakes, and humans are meant to make mistakes. And yet when we go to the serious adverse reactions every week, I'm seeing conditions that are sending alarm bells in my head, like anencephalic babies being born. So my question to the MHRA was, has this been reported as a result since the vaccination? How is this on the serious adverse reactions at all? Because you're talking about anencephalic babies, you're talking about stillbirths. We're also talking about, many people are talking about periods in women who have both taken the vaccine and those that haven't taken the vaccine. But I noted back when Brian and I started talking about all of this, that one of the big serious adverse reactions was vaginal hemorrhaging. And yet at the start of this, we were only vaccinating the elderly, 70 plus. So how was that happening? And then we're getting data that's not being input. We're getting the MHRA admitting that they're making mistakes. We don't get any detail with regards to the serious adverse reactions, unlike VAERS in the USA. We don't get told whether it's one person suffering a number of symptoms, whether that person is male or female, how old they are, what was the environment or the circumstance with which they experienced a serious adverse reaction. And when we link anything to the vaccine, the government and the general narrative is very keen to come back with, well, we cannot prove that this serious side effect was anything to do with the vaccination. However, my counter challenge would be that we're not doing the forensic examinations and the postmortems in order to definitively say that. So we're getting some very, we're getting psychiatric serious side effects now. We're getting pretty much every system in the body. And if someone has a pre-existing medical condition, then many of them are finding an exacerbation of that medical condition after they've had the vaccination. So we're finding data that's appearing and then it's disappearing, and it seems to be made up as it goes along. It's exaggerated or it's underplayed and ignored, but the truth definitely isn't coming out, and accurate information that we can trust, I would say 100% we're not getting, as UK column have shown many, many times before.
So perhaps I can come in and ask, are we unique in this? Are we seeing something special happening in UK? We're being suspicious. Are you seeing these things happening in France or Luxembourg or other countries where it appears that the statistical presentation by the government has got some weaknesses?
I know that the VAERS, the US-based vaccine adverse reaction system of the USA, has also problems in misrepresenting the data that disappear, especially dead people and so on. I don't know.
Under-reporting as well, Harvey. I should have said, just to include, because I know that you've done a lot of valuable work in that area, and I know Anne-Marie has noticed differences too. We're seeing a huge under-reporting of serious adverse reactions through the UK scheme, which is the yellow card scheme, which I know differs from your scheme. But the under-reporting is a major fact, we believe, as well. Sorry to interrupt.
Yeah. So people are saying that only 10% of adverse reactions are being reported by. And I know, for example, because but among them, among the adverse reactions, there is a paralysis bell with Pfizer. There are clots with AstraZeneca in March, which that's why the campaign has been stopped in 14 countries, among them Iceland, where somebody dropped. And we had a very big cases of in France of students of medicine. He was 25 or 26 years old. In 6 years of medicine, he took the jab, and then 2 weeks after, he got a heart attack, and they found him dead. And he was very sporty, very healthy. He had no comorbidity or whatever. And so people, I think, are, and I heard about Guillain-Barré being reported, or clots in the brain, like pulmonary embolies, and also clots in the brain, like stroke. And now we are hearing about myocarditis. And so these are very severe reactions, I think. And as everybody has said, it seems that it's a very underestimated, and there is a reason why, because they, for a long time, they forbid autopsies to be made on bodies, arguing that it was very infectious. So, for example, the people in houses who were dying from COVID, there were, for example, in France, they were being denied funerals, and they were being brought to how would you say, to Rungis in Paris. So basically, it was very weird. Like people, their family couldn't get the body, couldn't do the funerals, and the physician couldn't do autopsies. But I think that there was physicians in New York and in Italy who went against the guidelines and who did autopsies of the bodies anyways. And that's how they find out that it wasn't pulmonary, normal pulmonary infection, as they made us believe first, but it was due to a shortness of breath, due to the fact that there was clots forming in the arteries, the pulmonary arteries. And that was the first thing that people said, oh, it's not a pulmonary infection. It's not. So that's I think a lot of things are being hidden from us. And also Dr. Ox, he told me that he has to report, like, all the practitioners who belong to a network of Eurovigilance, and they need to report secondary effects. Like, he has been reporting a high level of D dimers, a normal level of leukocytes. It means no infections. So that's weird, because if it will be a normal virus, you should expect infections, but here there is no infection. So and he has a normal level of a high level of CPR. So we discussed with Dolores Cahill, and she told us that the D dimers were the product of the result of the product of degradation of the fibrin. Fibrin, when you have a clot, normally, like when you bleed, so the body do a clot by sending fibrins and then platelets. And the combination of the two, fibrin and platelets, make the coagulations clots and avoids any hemorrhage. What we see is very different. It's because some people have been reporting clots, but without platelets. So yes, it's an inflammation process, but it's not. It's another process that is being here involved. And this is my understanding, that I read that sickle disease, it's a genetical disease. You have these symptoms. It means you have no infections, but you have no platelets, and the coagulation comes because of a protein which is present of the surface. It's called the P-selectin, and the P-selectins make an aggregate of with the low, and then you here you have, because it plays a role in the adherence, and here you have your clots. So you have these clots, but without platelets, and that's what puzzled a lot of physicians. Harvey sent me a video of a woman, a physician, an MD, who had this problem. Like a woman came to her office and she had the clots in the veins. So clots in the veins, in the brain, at the back of the brain. But as she didn't want to put her on heparin, on aspirin, anticoagulation, because she was wondering about this absence of platelets. So my understanding is they use PRCAS9 editing gene that they inject with the vaccine in vivo, which is able to modify the change on chromosome 6, this amino acid, which is a glutamate, and change it into a valine, and this prevents the shape of the blood, and the red cells is unable to fix the oxygen. So here we have those symptoms of shortness of breath, but it's not. It's partially due to the inflammations of the lungs, but it started and it has been triggered by the formation of these, and that's what they want to trick us into believing, that it is an inflammatory process due to the infections. But it's not only that. It's not. It's much more complex than that. Much more.
Right. Okay. I'm going to suggest we come back to that. I want to just draw you back a little bit as well, because you started to talk to us about what was being done when people were presenting with COVID-19, and you said that they were being given paracetamol and effectively sent home, and then they were being brought in and put on oxygen. And you said, this is wrong. This is wrong.
Yes.
Can you tell us just very simply, take us through what was happening with people and why you believe this is wrong?
It's wrong because there have been research scientists publications showing that the paracetamol was triggering the inflammation process. And they said, oh, we should not use paracetamol. Right, and if people are being sent from day 1 to day 5, they have these symptoms. So they have fever, their nose is maybe running, and they can't swallow, and they can't smell, and then they go to see the physician. Day 5 to day 12, they are given a box of paracetamol and they are sent home. But from day 5 to day 12, what happens is that the body enters into an inflammation process because of the S protein. Because once the S protein enters into your body, it is it attracts APC cells. It means, for example, macrophage or dendritic cells, which are going to come. They're going to take in the S protein, for example, or the whole virus, and they're going to process it, and then they're going to present it on the surface to lymphocyte T, like CD4 or CD8, and it will trigger a whole cascade of signaling, if you wish, through interleukins, which is going to say to the cells, come, there is a virus here who is attacking the cells, and the cells are infected. The cells are producing and replicating the virus. So you need to tell the T cells or the B cells to come and to attack the cell which are infected, those cells, or to secrete antibodies that are going to kill the infected cells. But during this process, there is a process also of inflammations where the immune system is calling other cells like eosinophil or etc. So those cells who are responsible not only of the inflammation process, but also on the allergic reactions, like the anaphylactic reactions. So, and this is happening between the 5th day to the 12th day. That's why they are telling people to quarantine 14 days, because if your body recovers between 12 days and 14 days, you're fine. But if not, it means you're going to die because of organ failure and because of death of the cells, of course. So that's why it's critical not to let evolve the disease from day 5 to day 12 and to treat it like Dr. Ox is doing, and also some other physicians, Dr. Raoult, with anti-hydroxychloroquine, which is an anti-malaria drug, but it's also used to treat autoimmune disease such as rheumatoid arthritis. So it's dealing with the anti-inflammation process. It's blocking the anti-inflammation process from on. That's why it's important at the beginning of the disease, between the 5th days and the 12th days, to administrate hydroxychloroquine and then toxin, which is an antibiotic, to prevent sewer infection. Because once your body is getting weak, your immune system is not working properly, you catch up normal bacteria that is going to make you infected and causes sepsis. And that's another problem. That's why they give azithromycin and the zinc. Zinc is boost your immune system, like vitamin C, right, or vitamin D, or is boosting your immune system. So it's very important if you don't want to end up with severe symptoms and long COVID disease and going through these intubations where through oxygenations where you have 1/2 chance of survival, like of survival, to treat the disease from day 5, because that's when people realize that they're getting infected, to day 12.
Okay. Debbie, just help me out a little bit here, because Anne-Marie's taken us through that description of what should be happening, but we've had senior government officials here in UK encouraging people to be taking paracetamol as if it was the right thing to do.
Yeah, that would be Professor Jonathan Van Tam, I think you're referring to there, Brian, who actually stood at the podium in Downing Street, I believe, and lifted up a leaflet of paracetamol and said that everybody should get the injection because the side effects from the injection were no worse than in a box of paracetamol. However, paracetamol is one of the most dangerous drugs actually on the market, and that's why if you go in the UK to a chemist, they'll only let you have one box at a time. And it does have some very serious side effects if you've got any underlying conditions. And there are some recommendations for not taking paracetamol. And what I've been fascinated to learn since talking to Anne-Marie is that actually what we've been hearing about, what the public have been hearing about, is something called Cytokine Storm. And it's become kind of a bit of a buzzword now, and everybody talks about it, but nobody quite understands what it is. And what it is really, in very simple terms, is the immune system going into meltdown and starting almost to attack itself. In fact, it goes into panic mode. It's like dialing a 999 for an emergency ambulance, and the immune system's panicking so much. And all of these things are happening in the body all at once that you end up with almost a perfect storm. And that cytokine storm results in multiple organ failure. And of course, multiple organ failure will often result in death. But what they're saying now, and what experts like Anne-Marie are saying, is that paracetamol, if it's taken in the early days, can sometimes enhance and induce and exacerbate a cytokine storm. So, I mean, I'm not saying that there are grades of cytokine storm here, but maybe if somebody was going to experience cytokine storm, they might experience a grade 2 by the Met Office, for example. Whereas if they've had paracetamol prior to being, or at the early stages, then the likelihood is that cytokine storm will be far more severe and will need emergency treatment, as Anne-Marie was saying, with mechanical ventilation in an intensive care unit, and oxygen therapy, which I'm not sure at the moment was probably the right way to go, but then that's all up for discussion as well. So cytokine storm and paracetamol aren't a great mix.
Yeah. Right.
What people are recommending is, if you've got a medical condition that allows you to, and some people aren't able to take aspirin, but actually to take aspirin if your medical condition allows you to, rather than paracetamol. Am I correct in that, Anne-Marie?
Yeah.
Aspirin as opposed to paracetamol, or Tylenol as it's known.
If you don't have hemophilia problems, because some people are having hemophilia problem, it means they aren't able to coagulate, so they are forbidden to be given aspirins. But that's a very small percentage, I think. But most of us can take aspirin. Me, for example, I take regularly. I bought some aspirin tablets, like to chew. And because I saw a physician who said that you should, if you experience like shortness of breath and before it gets worsened, so I simply chew gram, for a couple of days, like 3 or 5 days, and then I stop, and it goes away, and then I am good,
Right.
It's probably also worth noting, Brian, that if anybody wants to go and look it up, you can actually make your own hydroxychloroquine at home with the juice of, I think it's oranges. It's a very simple recipe, but you can make a hydroxychloroquine homemade, sort of, if you can't get hydroxychloroquine. And just to reiterate the importance of zinc as well, you can pick it up in the UK over the counter, just like anything else. And I really would advise everybody to pick up some zinc. It will help. And vitamin C and vitamin D, obviously, too.
And you can't drink, so a tonic water, like, I don't want Schweppes, but like Schweppes, I don't want to say a brand, but it gets quinine in it. So you can't drink those tonic water. That's what I did during the first. And you can also make yourself, cook yourself some anti-inflammatory drinks. For example, you take 1 liter of water, and then you put make it boil with like 10 grams of ginger or with 10 grams of curcuma, and then not boil it because it's straight. And then you put one lemon juice, and you can add some pepper or some chili that will it's a vasodilatator, so that it prevents, and it's prevents the cytokine storm. So a lot of people are now looking for like drinks to prepare themselves to stop that, and it's natural. It's like you can drink this drink, and it helps you. Like curcuma is very antioxidant power.
Okay.
So, yes.
Let's come on to that maybe a little bit at the end, and we can leave people on some sensible things that they can do.